solution related to proteomics, genomics, and also flow cytometers and cell biology. So in case you are interested in any type of molecular biology research topic, so we can try to fit your requirements according to our different solution. And in terms of regional coverage, we work with our partners who is LASEC, responsible from the South Africa, in also main Africa, and related to life science division, we work with a collaboration with them. It doesn't mean only they are providing sales, but they have certified training teams related to service. They have certified and trained teams related to training and application. So when you think about all of these advantages, we are really happy to work with them, and you will get the same quality service and application and support from their side as well. So let's talk about chromatography systems from Bio-Rad and how we can help you related to this. And as you know, NGC chromatography systems are designed by researchers from pharma and built by Bio-Rad. And we have different configuration according to your different needs. And I would like to give like general overview of modules that what we provide. Basically, we have a Quest configuration, which is called NGC Quest 10 Plus. And this system, as you see these red rectangles, we have system pumps. We have two separate system pumps and a mixer. In addition, we have injection valve. We have a multi-wavelength UV detector with conductivity. We have a touch screen, and we have buffer tray. These are the basic components of our system, and all other different configurations, we do provide the same pumps and same detector. So there are no differences related to these modules. We have, of course, offered different flow rates from 10 to 100 milliliter, but we can improve or we can add different modules depending to your needs. Like it can be inlet valves, it can be column switching valve, it can be sample inlet valve as well, and it can be integrated sample pump. So whichever, let's say, whichever type of experiment or purification you want to achieve, we can fit the best configuration according to your needs. For example, if you don't need to use any buffer blending option, and if you don't need to purchase any outlet valve, so you are flexible to reduce your configuration without using these modules. Accordingly, you will not spend more money and you will reduce your investment cost. As a unique advantages of this system, all of our configuration has touch screen control. Whichever configuration you have, it comes with default touch screen, and you can control all the modules. And when you run your system, you can control all the run parameters. In addition, we have point-to plumb. I will give a short example video related to how it works, but basically it works like we have LEDs in each module's inlets and outlets, and you will be able to see all the tubings between one module to other module that will be blinking with LEDs when you choose the selected part on the fluidic scheme. In addition, we have edit on the fly capability with remote control. So if you connect your system to a Wi-Fi router, as much as you add mobile phone or computer, you will be able to control your device remotely. Of course, we have a free of charge, which is not CFR Part 11 compliance. As you know, mostly research-related application doesn't require this qualified software. So accordingly, you will have free of charge software and free of charge updates related to this software. Accordingly, you can install the software to your personal computers or your office computers. Accordingly, you can create your method or analyze your results without attaching the computer in the lab, and it brings also a lot of flexibility. Of course, related to these systems, we do provide optional air sensors, but on the other hand, we do provide sensitivity on the air sensor control. Accordingly, you can allow really small, tiny air bubbles which will never interact to your column and will dissolve with pressure, but you can stop the pumps whenever big air bubbles recognized by the system. We have multi-wavelength detector, which is 4-channel from 190 to 800, and it's default for all our configurations. It also includes a conductivity detector, and it's the wide range UV detector comparing all. For blending valve is available for any configuration. So even you have, let's say, low-end system, I don't want to say it's low-end or high-end, because at the end of the day, you will have the same pumps and same detector, means that you will have the same quality results. Plus, we have additional valves, whichever you want to do. And of course, we have higher pressure tolerance comparing similar range of systems. It allows you to make your purification easier without reducing your regular flow rates in case you run a high pressure size exclusion chromatography. And we have flexible flow paths that we are able to reduce delay volume whenever you need to collect really tiny amount of sample. So you can achieve using this flexible flow path option, we can reduce flow path. Accordingly, we can reduce delay volume. And the most important part, we have ready templates for any type of chromatography, including affinity, size exclusion, ion exchange, chromofocusing, in addition to 2D or tandem chromatography as well. So this is one example of how our chromatography systems has this touch screen and plus point-to-plumb control. As you see, when you run your systems, you will be able to see all the plumbing part of the modules with different lights. So green lights means you have some buffer line, and this blue light shows your sample line, and you will be able to control all the part from your touch screen. Of course, these advantages brings a lot of benefits. So we have really fast installation option that we can install this system under 4 hours. It doesn't take several days. Of course, the system has really easy to use and easy to learn software. Accordingly, you will have less training needs, and you will be able to have option of fast startup. And because of this flexibility, you will have less investment, and accordingly, whenever your needs increase, you have plug and play module, and you can upgrade your system according to any configuration. Because of these additional options and the flexibility, you have less technical support requirement. And as I mentioned again, the same pump and same detector for all configuration provide the same quality results for any application. And I will give one example related to this. And all of this provides faster optimization for the method and process development at the end of the day. So this is an example from our NGC brochure. So it's comparing two different systems, actually 3 different systems. I would like you to focus these blue lines with purple and orange one. So as you see, this is an analytical column, which is size exclusion column, which run with using gel filtration standard with different molecular weights. I would like you to focus the peak points of these standards that you will be able to see. These are almost identical. So these two different systems using the same column and same samples gives almost the same results. So accordingly, you can directly transfer any application from different chromatography systems. So how we can optimize our workflow or we can customize our solutions. As you see, real flexibility in action is our most advanced point, let's say, because let's think about protein A affinity chromatography for IgG purification or monoclonal antibodies. As you know, there are several steps. Like in that case, there are 5 different steps you need to proceed to get the purified samples in lab scale. And it requires a lot of hands-on time. It requires a lot of patience. It requires a lot of focus to purify that proteins. So in that case, we are really flexible to make this purification automatized. So accordingly, we can make a custom tandem purification. So we can collect the protein in protein A, then we can directly dissolve them. After that, we can collect the purified proteins, dissolved proteins in a loop. Then we can send anion exchange to, then after that, a cation exchange or one of them. Then we are able to collect in another loop. After that, we can use the high resolution size exclusion column and we can collect the fractions. So as you see, our system allows that we can include 2 buffer inlet valves. Then we can include 3 column switching valves, that 2 of them for column and 1 for loop. Plus we can collect even a lot of sample using 2 NGC fraction collector at the same time. So it brings a lot of flexibility. So according to your real needs, we can configure these systems. There is one example from Sanofi, which is published before, and they use similarly same experiment. So this is the experiment using 5 different cell culture supernatant, and they apply 3 different steps of chromatography, which is protein A affinity, then gel filtration, then dissolving. During that steps, there is no fraction collector in between gel filtration and protein A and dissolving. So at the end step, last step, which is the gel filtration column, they are able to collect their fractions. So we are able to share these documents to you if you want to see the details. Another good example comes from Amgen. So they also try to purify antibodies, and they try to load like challenging amounts, like from 200 to 500 milligram of the antibody concentration. And at the end of the day, they will be able to purify high yield of proteins, antibodies, comparing the competitor system. So this publication is also available from our side, and we are able to share it to you. So let's talk and the others, because it's not only related to the system, also related to columns and resins. According to, I will give a slide related to which type of columns and resins we do provide, but we are able to provide a purification solution for plasmids, viral vectors, viral-like particles, vaccines, IgGs, recombinant protein, native proteins, any monoclonal antibody you may think about. So Because we do provide different type of resins, like these are ion exchanger, anion and cation exchanger, affinity resins, mixed mode resins, which I will give several examples related to it that will help you to reduce your steps. Instead of 3 steps, you may go to 2 steps, or even you can capture your proteins in single step instead of 2 or 3 steps. So first resin is highly important because, as far as I know, many researchers interested in recombinant protein purification, and this resin has highly binding capacity, dynamic binding capacity, which is 40 milligram per ml resin. And you don't need to recharge the resin after several purification. As you see, after 100 cycle of repeated purification, there is no need to recharge the resin. And this resin also has high shelf life, like 5 years. This is the really robust resin in case you are interested in histidine tag protein purification. On the other hand, in some cases, when you produce recombinant protein, you are not able to add tag. In that case, we have 2 examples, like non-tag recombinant protein purification directly from the fermentate, which you may be interested in. One of them, we are using Nuvia A'4A that allows us to capture the recombinant protein almost 80%, with the recovery in the first capture more than 75%. Then we are able to use the other mixed mode resin, which is a ceramide hydroxyapatite, and we can go to purity more than 95%. And we will be able to share these technical notes with you as well. The other examples, I would say, again, non-tag recombinant protein purification, which is this is the Nuvia C' resin, and we are able to purify this raw starch digesting alpha amylase. So in that example, we directly load this feed batch culture. Then we are able to capture almost 90%, 96% of yield of the enzyme, which is still active units. The other example that I mentioned in previous slide, automated monoclonal antibody workflow, that we can convert to automated histidine tag protein workflow as well. Just we need to modify the resin, which is here protein A, but we can change it to Nuvia IMEC. So accordingly, we can capture histidine tag proteins. But in this example, as you see, the steps takes longer when you think about multiple steps to get expected purity. But when we make a custom solution for you, actually, it's already, the method is already in the software, so which is called tandem chromatography, or 2D chromatography, which is here multi-D. So just you need to choose your resins, and these resins, of course, and buffers should be compatible to each other. But in this tech note, you can see the details, and we are quite flexible to optimize loading amounts, buffer and washing times, plus fractionation. So you can customize these methods easily. The other example, I would say, like CGMP ready purification process for adenoviruses, which is highly important for nowadays CAR T cell therapy. And we have really unique resin which can capture high molecular weight proteins mostly, and viruses in single shot, which is called Nuvia HPQ. And this is an example of how we scout different type of resin to capture these proteins and viruses. And you will see Nuvia HPQ has unique binding capacity and recovery as well. And you will see in these examples, stability and reusability of this resin is quite good even after 100th cycle of purification. So in the last part of my presentation, just I would like to give brief information related to protein purification workflow, because in any which type of protein you are interested in, you need to apply different steps related to your protein's properties and expected purity levels or expected amounts. At the beginning, you will have a bulk protein from lysate or supernatant or any part of isolation. Then first, as you see, we need to capture this protein. Any type of capturing step is possible, as you know, depending to protein's properties. But after capturing, most probably we need to run a quick STS page to see the results. Then we need to decide if we need to go for intermediate purification or polishing step. Means that you may go for 2-step or 3-step purification, and each of these steps you need to run solution and comparably fastest one. But at the end of the day, when you try to cast your own gel and run the gel, then stain the gel, even you may use fluorescent stain, or mostly you are going to use commercial staining. It should require overnight staining, but it's not enough. You need to make really proper destaining to see your results, in case you need to see really low amount of contaminants. So in our workflow, you don't need to cast a gel. Of course, you are able to cast a gel, which is stain-free gel, using stain-free fast cast gel casting solutions that I will give these examples to you, or you can use precast gel. And using our criterion system, you will be able to load almost more than 18 samples, up to 26 samples, and you can run these gels these proteins in 5 minutes. Again, we are able to provide these tech notes to you. So accordingly, when you run your hand cast gels, which is cast by our provided fast cast solutions, these are stain free, or when you run precast gels, you can run up to 26 samples in the single gel, and just after you load all of these fractions, you will be able to see results in 5 minutes, which is highly sensitive, almost sensitive as silver staining, and it's really helps to get your results as quick as possible. So my presentation is finished here. I would like to get any question you are interested in, and I would like to respond as much as I can. Thank you for listening, and I'm really appreciated.